Hello everyone, myself Monu from group number 322. Today our topic is the kinetic property of muscles. The quantitative theory expounded above refer to study muscles contraction. Of no less interest are the dynamic property of the muscles under non-steady state condition. The steady contraction has the character of plastic flow. In non-static condition, uh, the muscle exhibit elastic property. For instance, in quick release experiment in which the isometrically contracted muscle is released and experience rapid isotonic contraction, there are observed slow damped oscillation. The frequency of these oscillations is of order of kilohertz for muscle several centimeter long. If we regard the muscle as a viscoelastic body, we can construct a model containing a damped elastic element connected in series without an undamped elastic element and one more elastic element in parallel with the first two element. Such a formal model is combi- combination of Voigt and Maxwell bodies. The Voigt model in the elastic element connect in parallel with a damped element, and the Maxwell model contain the same element connected in series. The generation of oscillation in the muscles can be determined by the non-linearity of non-steady state kinetic equations that do not contain elasticity in an axillate form. The possibility of oscillation in is in this case due to kinetics of closing the and the opening of cross bridge. On the other hand, the bridge itself is a viscoelastic system. The tension generated by a closed cross bridge may vary in a stepwise manner depend on the angle at which the head of HMMS1 arranges itself relative to action actin, and also on the extent of stretching of S2. The transition between the step have an effect on the rapid non-steady response of the muscle. Thus, the oscillation during quick release are caused by the elastic deformation of the bridge. This is the most probable through not yet proved supposition of great interest for physics and biology are the fight muscles of insect and the relative tympanal muscle of Caestus. These muscles are capable of rapid periodic contraction with frequency of the order of 100 Hz. The fight muscles of insect are structurally similar to the cross-threaded muscles of the vertebrates. It has been established that the sliding filament model with actin myosin bridge is applicable of IFM. The rapid oscillation of IFM required the presence of a directly functioning elastic element microscopy so that in contrast to the muscles of vertebrates in IFM there is a direct viscoelastic link between myosin filament and Z membrane accomplished by a special element C. Probably this element is capable of undergoing elastic deformation. Uh, there has been stabilized the large difference between the oscillation of IFM and those of the potential transmitted to these muscles. For example, the frequency of the potential transmitted to IFM is 3 Hz and the frequency of winning oscillation reaction is 120 Hz. Hence, the muscle is not activated with these oscillations. The oscillation of IFM are the nature of auto oscillation. These auto oscillation states arise in non-line non-linear system due to the focus dependent on the state of movement of the system itself. The amplitude of auto oscillation is independent of the initial conditions. The auto oscillation of IFM are exceeded in the presence of a feedback between deformation and stress. The relationship between these varies depending on the active state of the system. Presumably, IFM contain a transfer element which responds to mechanical event and control the state of the contractile system. This element is localized in myofibrils, which is proved by the presence of auto oscillation in IFM preparation boosted by glycerol. Thank you.